Hello, I'm Luca Rettenberger, and I'm presenting you a quick conclusion of our most recent work here at the Institute for Automation and Applied Informatics within the Karlsruhe Institute of Technology. With me, I mean Marc Schutera and I, as well as Christian Peilatek, who contributed the data we used for this work, and Markus Reichel, our academic advisor. First of all source code and data is open source and available online. The source code is written in TensorFlow and Keras, and the Jupyter Notebook is online ready to be executed. The data is hosted on OSF and can be downloaded there. If any questions remain open or further assistance is needed, feel free to contact Mark or me. All links are also provided in the video description. In this work, we use images of medaka hatchlings. This is a type of fish which is see-through, which makes it non-invasive studies regarding the internal organs and the whole circularity systems practical. Each sample is an RGB image capturing the heart region from a constant ventral view. In each sample, all three heart chambers of the fish are visible. For each sample, the segmentation of all three chambers is given. In the sample below, we can see that the ventricle here is segmented in white, the bulbus in black, and the atrium in green. In total, there are 565 train samples and 165 test samples in the dataset. In our work, we realized that, especially within biomedical imaging, datasets are very small scale. This is due to biomedical data often being very tedious to label and most of the time need an expert to provide additional labels. This problem is even amplified within semantic segmentation, since providing segmentation masks is often more difficult than simple classification labels. The combination of those problems often lead to unclear datasets. This means that the dataset may be labeled heterogeneously, which means that for each sample different labels may be provided. In the example below, we can see such a situation. We have 4 samples, each having different chambers of the heart segmented. In the first example, we only have one chamber segmented. In the second and third image, we have 2 chambers segmented, and in the last image, all 3 are labeled. If one now wants to work with such a data set, there are essentially 2 options. On the one hand, you could label the whole dataset for it to be homogeneous, so every sample has a label. This will cost much time and effort. On the other hand, you could throw away samples which do not have all classes labeled. You could, of course, do that, but much information will be lost. In our work, we try to tackle this problem by firstly providing a fully labeled homogeneous benchmark dataset of the medaka heart region. This means that for each sample, all 3 heart chambers are segmented. Then we provide a novel objective function which does not only cope with missing labels for certain samples, like all other works already did, but fully leverages implicit information with partially labeled samples. Lastly, we conduct multiple experiments for realistic settings one may encounter. The objective function used in this work is tailored to enable learning on heterogeneous labels and stabilize the learning process by exploiting implicit information of unlabeled classes. The available class labels for each sample and the predicted output of the neural network for each pixel are utilized when calculating the objective function's output. Our combination of 2 objective functions consists of 2 individual objective functions. On the one hand, we have the modified dice loss, which is responsible for the simple channel-wise optimization, as we already know it in classical semantic segmentation. On the other hand, we have the class asymmetric loss, which does distribution-wise optimization. In the image, an exemplary sample X is fed through our used segmentation network. It outputs a training prediction Y tilde. Each class here has a unique color for the segmentation. The corresponding ground truth label Y hat is missing the class on the lower right, which is denoted with a dotted line where it should be. The dashed box on the right is the combined objective function. Prediction errors are given in red within each of the loss functions. The dice loss will output a value greater than 0 for the two given classes and 0 for the unlabeled classes, since it cannot make a prediction when no labels are given. The class asymmetric loss will only output a value greater than 0 if the unlabeled class's predicted segmentation masks intersect with a given class's background truth mask. Since the network predicted a small portion of the unlabeled class as part of the class in the center left, the output will be greater 0 for this class. For the other labeled classes, no intersection occurred, and hence the output is 0. For the evaluation of our experiments, we introduce a novel measure, the performance frugality ratio, short PFR. It measures the similarity between ground truth labels and predictions under consideration of the degree of heterogeneity in the given data and an existing statistics, which will be the intersection of a union in our case. So the PFR divides the YOU by the number of available labels. Since PFR takes heterogeneously in the dataset into consideration, the calculation is more profound than the IOU alone. For details about this, see our paper. So, the first experiment focuses on the central claim of our work, to demonstrate the ability to train on heterogeneously labeled data. The effect is presented on different magnitudes of heterogeneity, so labels from all classes are dropped equally with an increasing percentage. This is no specific scenario, it is just a general insight on how missing labels influence performance. It also provides the baseline for the other experiments, since if we drop 0% of labels, this means we have a fully labeled dataset. And with this experiment, we can see that up until about 60% missing labels, no significant drop in performance can be observed. So that means that more than half of the labels can be missing without loss in performance. This is quite an astonishing result, since it means that in the case of our dataset, most labels obviously do not introduce much more information and are pretty obviously not really needed. For the second experiment, we only evaluated two classes, the ventricle and the atrium class. In the second experiment, a situation is assumed where the dataset is fully labeled with a single class and should be extended by a second one, so a quite common situation. For our case, this means that the ventricle is always labeled for every sample, and the atrium class contains a decreasing number of labels to simulate an incomplete labeling process for the atrium class. This experiment shows that only about 30% of the ventricle class need to be labeled to achieve almost as good results as if the whole dataset was labeled. This again is quite astonishing and means that if one wanted to extend our dataset in a real setting, it would not need nearly as much as all samples to be labeled. The third experiment simulates a situation where two datasets are sampled from the same domain but with differently labeled classes. Here, two datasets containing the labels for distinct classes should be merged without additional labeling. So there is not a single label where both classes are labeled in this case. For this, the initial dataset is split into two distinct datasets, one only having the ventricle class labeled and the other one only the atrium class. The synergy for different splits are then evaluated. The results show that the best performance can be observed if the two datasets are balanced, which is to be expected. But slight imbalance is also feasible and achieves good performance. This means that datasets from the same or very similar domains can be merged without any labeling needed. The last experiment investigates how the two individual loss functions influence the performance. For this, alpha is used, which weights the individual terms of our loss function. The weighting factor alpha is modified to see how both loss functions influence the learning process. The best performance can be observed at alpha about 0.5, which means slight more importance on the dice loss. The best performance can be achieved if the two losses are employed at a roughly equal share, even though there is a slight more importance on the dice loss. This work provides the deep learning practitioner with a method for supervised learning on heterogeneous labeled data. In particular, the method's contributions to frugal label deployment is of interest to the biomedical research and engineering community, which is often faced with multiple challenges addressed in this work. The dataset, the entire training pipeline, including the heterogeneous dataset generator and the proposed objective functions implementation, are publicly available, and the links are provided down below in the description. Thank you for your attention.